In this demonstration, we are going to observe the general reactions of carbohydrates. Here we can see the monosaccharide solutions glucose and fructose, reducing disaccharide solution lactose, non-reducing disaccharide solution sucrose, and the polysaccharide solution which is starch. So this we can see it is partially soluble colloidal solution compared with the monosaccharides which are completely soluble. Here we can see the reagents. First is a Molish reagent which is used for the Molish test. The Molish test is a test to identify the carbohydrates. Iodine solution is used in the iodine test. So iodine test is used to identify the polysaccharides. Benedict's reagent is for the Benedict's test. Benedict's test is a test to identify the reducing sugar. Barfoed's reagent is used in the Barfoed's test. Barfoed's test is used to differentiate between the monosaccharides and the disaccharides. Seliwanoff's reagent is used in the Seliwanoff's test, which is used to differentiate between the aldose and ketose sugar. The last one, the phenyladrazine hydrochloride reagent, is used in the Osazone test, which is a confirmatory test. First one is a Molish test, which is a test for carbohydrates. For this, 1 ml of the sugar solution is taken. To this, 2 drops of Molish reagent is added. Then we have to add 2 ml of concentrated sulfuric acid along the sides of the test tube. We have to tilt it slightly so that it will form 2 layers. Appearance of reddish violet ring indicates the presence of carbohydrates. Iodine test is a test to identify polysaccharides. Here we have the fructose and lactose solutions, which are monosaccharides and disaccharides respectively, and the polysaccharide starch. Here I am doing the iodine test in the sugar solution fructose. For the iodine test, 2 ml of the sugar solution is taken and 2 drops of iodine reagent added. Iodine is brown in color. Here we can see there is no change in the color of the iodine solution. For the starch solution, 2 ml of starch solution, I am adding 2 drops of iodine. Here we can see an intense blue color formation. This shows this contains the polysaccharide starch. Glycogen forms red color with iodine solution. Benedict's test is a test to identify reducing sugar. Here we have the monosaccharide solution, reducing sugar solution glucose, and non-reducing sugar solution sucrose. So for the Benedict's test, we need to add 3 ml of the Benedict's reagent and 5 drops of the solution. 5 drops of glucose solution I am adding, and we have to keep in the boiling condition for 2 minutes. The same way, I am adding the 5 drops of the sucrose solution in the Benedict's reagent and we keep in the boiling condition for 2 minutes. Reducing sugars are positive for the test. The appearance of the brick red color precipitate indicates the presence of reducing sugar. So we can see the change in color. First it changed to green, yellow, and finally brick red color precipitate. Whereas in the sucrose solution there will not be any color change. So we say reducing sugar reacts with the Benedict's reagent to form the brick red color precipitate. The reducing sugar solution glucose reacts with the Benedict's reagent and form the brick red color precipitate, whereas the non-reducing sugar sucrose is not changing to any other color. The Benedict's color is blue, it remains the same. It is negative for the test. Barfoed's test is a test to distinguish between monosaccharides and disaccharide. Here we have the monosaccharide solutions, glucose and fructose solution, and disaccharide, lactose and sucrose solution. For the Barfoed test, we have to take 2 ml of the Barfoed's reagent, I have taken already, and 2 ml of the sugar solution, that is a monosaccharide solution. The fructose solution I have taken. Mix well. So in the same way, in another test tube, we are taking the disaccharide lactose solution 2 ml and 2 ml of the Barfoed solution, which is added already. For boiling time is 2 minutes. So this will keep in the boiling water bath. Presence of the red precipitate indicates the presence of monosaccharide. After getting the precipitate, we have to cool in the running tap water. So it will not be so quantitative like how we get in the Benedict's. The precipitate formation will be very scanty. So only after cooling under running tap water, the red precipitate will form nicely. After 2 minutes, we can see the red precipitate formation in the monosaccharide solution at the bottom of the test tube, whereas in disaccharide solution, no color change. The Barfoed's blue color solution remains the same. Seliwanoff's test is a test to distinguish between the aldose sugar and ketose sugar. Here we have taken the glucose solution, which is an aldose sugar solution, and fructose, which is ketose sugar solution. To do the Seliwanoff's test, 1 ml of Seliwanoff's reagent is taken and 1 ml of the sugar solution is added. So in the same way, for fructose also, 1 ml of Seliwanoff's reagent and 1 ml of fructose solution is added and kept for 1 minute. The appearance of cherry red color indicates the presence of ketose sugar. Seliwanoff's test is specific for ketose sugar, so the fructose solution will give cherry red color in the positive Seliwanoff's test. Here we see after 1 minute of boiling, in the glucose solution there is no change in color, whereas in fructose solution, which is a ketose sugar solution, there is cherry red color formation. This is the positive color for Seliwanoff's test. Phenylarazine hydrochloride test or Osazone test is a confirmatory test in sugar analysis. It is called phenylarazine hydrochloride test because it contains the chemical phenylarazine hydrochloride. It is called Osazone test because the product formed is Osazone. Here we have the sugar solution, glucose, fructose, maltose and lactose solution, each 1 ml of the solution. In the phenylarazine hydrochloride test, 1 ml of the sugar solution, 1 ml of the phenylarazine hydrochloride reagent is added. So we have already taken 1 ml of the sugar solution. To this 1 ml of the phenylarazine hydrochloride reagent and kept in the boiling condition. In the same way, 1 ml of the phenylarazine hydrochloride is added in fructose. 1 ml of the phenylarazine hydrochloride reagent is added to the 1 ml of the maltose solution. 1 ml of the phenylarazine hydrochloride is added to the 1 ml of the lactose solution. Different sugars form, it's mixed well and kept in the boiling condition. Different sugars form different shape of the crystals at different time intervals. Here we have the ozone crystals formation for the various sugar solutions after the boiling for the particular timing, which differ from sugar to sugar. Here we have the glucosazone, fructosazone, maltosazone and lactosazone. Glucosazone and fructosazone will form the same needle-shaped crystals, only the time variation. Fructose will form the osazone much earlier than the glucose. Fructosazone will be formed within 3 to 5 minutes, glucosazone in 8 to 10 minutes. Maltose will form the crystals in 20 to 25 minutes and lactose in 45 minutes to 1 hour. Maltosazone forms sunflower shaped crystals. Lactosazone or tennis ball shaped crystals. The crystals are viewed under the microscope.